So now we're going to look at the physical structure of deoxyribonucleic acid, better known as DNA. DNA is, as we have just seen, a double-stranded molecule from which all the information that is used to make us who we are comes from. Now, to really see how the structure of DNA is constructed, we need to unwind the two strands and separate them. When we do separate the two strands, we can see the basic components for each are virtually identical, but they run in opposite directions. Isabel will talk more on this later in the video. Each strand of DNA consists of many nucleotides bound together by phosphodiester bonds to form long chains called polynucleotides. I know, a lot of long words there, but by the end of this video they will all make sense, don't worry. Now, what is a nucleotide? A nucleotide is one of the structural building blocks of both DNA and RNA. A nucleotide has three main components: a phosphate group, a 5-carbon sugar molecule called deoxyribose. This sugar molecule is very similar to RNA's ribose. However, this ribose molecule is missing a hydroxyl group on its 2' carbon. And then finally, one of the 4 nitrogenous bases: adenine, thymine, guanine, or cytosine. So every nucleotide in DNA, there are many, differ only in which of the 4 bases is attached. How all these components are arranged is very important for how each of the polynucleotides are formed. A phosphate group is bound to the deoxyribose sugar molecule at either the sugar's 5' carbon or its 3' carbon. The numbering of the carbons is essential in describing the directionality of the strand of DNA. This same phosphate is also bound to another sugar molecule next to it at the alternative 3' or 5' carbon of the sugar. This bonding of sugars and phosphates is what forms the phosphodiester bonds I mentioned earlier. You might be wondering, how do the two strands of DNA connect? How do they interact? And the answer is all down to 2 words: base pairing. So, the 4 nitrogenous bases mentioned earlier, adenine, guanine, cytosine, and thymine, can be split up into 2 groups. Thymine and cytosine are pyrimidines, and adenine and guanine are purines. When bases pair with other bases from another DNA strand, they have specific partners. Pyrimidines always bind with purines, and we can go even further to say that specific pyrimidines pair with specific purines. In the case of DNA, cytosine pairs with guanine, and thymine pairs with adenine. These bases pair by a type of intermolecular bonding that we are all too familiar with: hydrogen bonding. It's through these hydrogen bonds created through base pairing that the two strands of DNA are joined. As well as hydrogen bonds, DNA also has pi bonds. These help to form the double helix structure that DNA is so famous for. The sharing of electron probability created by the pi bonds creates 2 different types of grooves in the structure of the double helix: the major and the minor grooves. The grooves act as binding sites for incoming proteins, needed for DNA reading and for replication. Proteins know which groove to bind to by recognition of specific base sequences present in these grooves.